Hello and welcome to today's presentation, Soul Food for Cannabis, Nutrient-Dense Living Soils. Today's webinar is being makers of shield disinfectants formulated with accelerated hydrogen peroxide. You will have the opportunity to submit your questions by typing them in the chat section. Submit your questions at any time, and we will be standing by to make sure they get answered during the live Q&A at the end of this webinar. So without any further ado, let's get started. I would now like to introduce Av Singh, Cannabis Cultivation Advisor at Fleming and Singh Cannabis Inc. Over to you.
Thanks. Thank you everyone for joining in, and a special thanks to the folks at Virox for inviting me to give this talk. And without, just I guess a quick little jump in to talk a little bit about some values and biases. I always do like to talk a little bit about how important this plant is, how sacred it is to so many people. Want to be honoring the feminine aspect of this plant. I personally like to acknowledge the importance of the legacy market, the subculture that was continued to grow during prohibition. As a sector, we're always trying to get better, and that in many ways we have to address some of the diversity issues that we have. We need to address the sexism and racism, and hopefully we can make a better sector accordingly. And then lastly, and probably most important to what we're going to be talking about today, we do like to acknowledge that this plant, the cannabis plant, has for millions of years developed a relationship with the microbes in the soil. And really, so that's kind of the basis of this talk, and why this matters is why are we so interested in actually growing cannabis in soil or in a living soil? And I thought that this paper that came out late last year really speaks to, I think, some of the challenges we're seeing in the cannabis sector as well. So in this trial, they were looking at a root microbe, and that was added on to address fungal issues. And what they found is that the newer varieties of the tomato plant actually had very little relationship with this microbe, and so they were not able to address the plant pathogen using this. Meanwhile, the wild relatives of the tomato plant responded quite well to the root pathogen, or to the root protectant, and therefore will be able to address the pathogen. So I think that's happening a lot in the cannabis space as well. And when we look at why that matters is really trying to understand the importance of microbes in the relationship to the plant. So just a very quick little synopsis of some of the things that microbes, and in this case a bacteria, can provide a plant. One of the things that plants typically do is that when they go under stress, they may produce ethylene. That production of ethylene will tell the plant to stop growing. What these microbes can do is essentially address some of that stress, and so they can produce chemical compounds that will inhibit the production of ethylene, and therefore the plant will continue to grow even though it undergoing some stress. So, for example, transplant shock, right? So we've all experienced transplant shock as you're potting up a plant, as you're moving it from indoors to outdoors. That's a lot of stress on the plant. Microbes can actually reduce that stress, and the plant can continue to grow through that. Other things that microbes do, they can produce plant hormones. So tryptophan can produce indolic acetic acid, which can help the plant grow. We know that it can do some nutrient cycling, making nitrogen available, making phosphorus available, solubilizing other nutrients. Bacteria, as well as other microbes, use terpenes as a language. So they're always producing terpenes as a way of communication between other microbes and between the plant, so that terpene production is possible. Another example of nutrient availability is making siderophores to carry iron. Microbes will also reduce iron, make it in a form that the plant can take up. And then there's also the induced systemic resistance. So the microbes can actually heighten the immunity of the plant. And so that's, I think, why if we as a society really want to get away from using a lot of synthetic fertilizers, if we want to start using more biopesticides than synthetic fungicides, we really need to make sure that our plants are in fact going to react to the microbes that they're being presented with. So specifically, what are nutrient-dense soils? So when we're talking about living soils, oftentimes they're equated to super soils. And as far as I know, the real terminology around super soils came out of the 80s and were probably developed, as far as I know, in the Kootenays. And then eventually we started seeing other recipes like Subcool's recipe, the Rev has a recipe, Clackamas Coot. So there's a lot of recipes online which are considered super soils and occasionally also referred to as living soils. And a soil, so I put little quotation marks around this. Soil typically is a mineral component, and that's your sand, silt, and clay. And then you'll have a small component that might be the organic material, say about 5%. And then typically half of soil is air and water. Now that's the traditional definition of what soil is. When we're growing in these, in a super soil, typically we're referring to stuff that's going to be a container garden or in a living soil bed or something like that, that's for indoor cultivation. And typically you'll have a base material that is traditionally been peat. More recently, we're starting to see a lot more cocoa being, or a mix of the two. So you'll have a peat cocoa coir mix. Another component is typically something that's going to create aeration. Perlite is one of the more common used products. Vermiculite also works well in terms of aeration, provides a little extra magnesium, works a little bit better as a wetting agent, or helps create maintain moisture content in the soil a little bit better. Some folks use lava rocks, the hydroton, the expanded clay pellets. I would also recommend something like a charged biochar. There's a lot of interest around buckwheat hulls, rice hulls. With rice, you might want to be careful. You want to do an analysis to make sure that there are no heavy metals. Rice can tend to attract a bit of arsenic. Cocoa chips are another thing and some people are starting to use like wood fiber as well. So there's a lot of options to create aeration. In these super soils, of course, we want a living component. Oftentimes that's going to be compost or vermicompost, worm castings, and occasionally you could consider insect frass perhaps as well. There's going to be some good protozoa typically in insect frass as well. Then there's the fertility aspects, which come in the form of organic nutrients, rock powders, and then biostimulants. And biostimulants, in my opinion, is probably the most emergent sector within the sort of fertilizer and supplement area. They're not, biostimulants are not directly fertilizers, but they contain ingredients that may stimulate the growth of a plant. And we'll talk a little bit more about that a little later. Oftentimes in these soils, you do need a wetting agent. A wetting agent just makes water wetter. It allows for a better spread of water within the surface. Oftentimes in an organic system, you'll use something like yucca extract, anything with saponins, because that can help break the connection within water molecules. And lastly, if you are thinking of a just add water soil, oftentimes you may want to add very specific microbes that may speed up the nutrient cycling. And just to go into greater detail around some of the fertilizers, I'm not going to get into specifics here too much, but for example, in nitrogen, you can have crustacean meals, the crab meals, blood meal, feather meal, soybean meal is an excellent source of nitrogen. In terms of phosphorus, bone meal and calphos are more typical of what you'd see in a container system, especially when we're talking about organic products. In terms of potassium, kelp meal works great. There's an organic form of potassium sulfate, 0050. Great opportunity to add a fair amount of potassium, as well as having some sulfur as well. Calcium, which I still would often say is the most underrated nutrient. You can find calcium in limestone, whether that's a dolomitic lime or a calcitic lime, depending on in my opinion, your clay content as well as your need for extra magnesium. Gypsum, if you're trying to kind of maintain the balance but still add some pH of the soil, but still adding a little bit of calcium. So that's another way of adding a bit of sulfur. Oyster shell flour is another ingredient that can add some calcium. It's a little slower to release, but the finer the flour, the more access you'll have. Magnesium, a critical component in chlorophyll synthesis. So things like sulpomag, langbonite, K-mag, or Epsom salts can work. Trace minerals, often looking at rock powders to do that, and glacial rock dust, basalt rock dust are very common. Green sand, another one. Dehydrated seawater, and to me, another excellent ingredient is of course, kelp. Biostimulants, as I mentioned, you have the acids like humic acid, fulvic acid, aloe vera and yucca extract, coconut water. All of these often will have plant hormones and other things to trigger an immune response in the plant. Fish hydrolysate, kelp, chitin are other forms of biostimulants. So one of the main things we want to do when we're trying to create these super soils is to essentially say that our goal is to make sure we're feeding the microbes. And it's slightly different than the concept. Originally, we would say as organic farmers, our role is to feed the soil and allow the soil to feed the plant. In many ways, what we're really trying to do in a sort of a microbe-centered nutrition program is we're trying to feed the microbes. We know that the plant communicate with the living soil to dictate what it needs when it needs it. We just want to make sure that the microbes have exactly what they need. And so that does mean that we really do want to make sure that we are balancing our nutrients. We just because we like something, we don't need to add it so that it's makes everything else out of balance. Balanced nutrients lead to higher Brix levels. Brix referring to the total dissolved solids in sap, good estimation of what is a healthy plant. And then so critical are, when we're talking microbes, it's a lot around making sure that we have a lot of our trace minerals and some of the micronutrients. So looking at things like zinc and molybdenum, copper, cobalt, iron, silica, these are, some of them aren't even considered essential nutrients, but they play such critical roles in the synthesis of other enzymes, catalysts for other enzymes, and in the formation of a lot of proteins. So some of you are familiar with the term crop steering, and it's probably a little bit difficult to do crop steering just looking at your super soil. However, there are techniques you can use to ensure that you are steering your crop to go from a vegetative to a generative phase. And in large part, that where we like to focus on is around our nitrogen source. So we all know that nitrate nitrogen is very good at creating vegetative growth, very important. It's going to be picked up in the roots, taken right up to the apical meristem, and you're going to get positive growth, which is great for the vegetative period. After a while, we want to see that nitrate nitrogen form be replaced with more subtle forms of nitrogen, in the sense of like maybe ammonium or urea, or in some cases, some of the best ways of getting nitrogen into the plant is through amino acids. And so we want to design our soil so that we have a little bit of nitrate nitrogen at the beginning, but then it goes into more complex forms of nitrogen. And similarly, we want to time the release of our products like phosphorus and potassium to coincide when the plant's kind of demanding these. And so we know that phosphorus is very important whenever the plant is looking for energy, and that's often going to be early on and then just around that transition between vegetative and generative, or the flowering phase. So we want to make sure that we're using, in this case, in the case example I have on the slide here, we're using microbes that are going to make the phosphorus a little bit more available at a timely way. So in this product, Biofuse, we have Bacillus Subtilis, a strain that can solubilize nitrogen and fertilizer, nitrogen and phosphorus quite well. The interesting thing about designing super soils is that over time, you are actually going to increase your EC, which is going to create some stress in the plant and be another way of steering the plant into generative. As I mentioned earlier, biostimulants, I think this is a category that's really going to expand within the fertilizer and supplement movement. We talked about a lot of these, so just to highlight, that you have the humic and fulvic acids, a lot of these plant growth promoting rhizobacteria, so the PGPRs, extracts like kelp extract, and then some other things like the fish hydrolysate and others. So the real crux of what I was to talk about, here are 3 points that I'm going to go through. The first one being, growing cannabis using living soils will not inherently increase the likelihood of inconsistent potency results or failed microbial testing. In fact, it could be quite the opposite. So the most important thing to remember is that you don't want to create the wrong growing environment, right? So ideally, we've got the environmental conditions in which we are having great transpiration. The plant is taking up a lot of water. With that, you're bringing up good amounts of calcium. Other nutrients are coming in very timely. So temperature, humidity, critical to growing that plant really well. Plant nutrition, also so important. That's where you want to have the balance. You want the plant to be able to dictate what it wants, when it wants it, and in what form it wants it. And then, of course, one way to really monitor your microbial loads is to make sure that you're sanitizing well and that your staff have proper hygiene. In creating that right environment, one of the things we really try to recommend with a living soil is that we move away from oscillating fans. Oscillating fans will often create non-uniform environments, greater opportunity for pathogens to work. And at times, if misdirected, they can tend to blow a lot of the soil away from the top surface and blow it onto the leaf material. So we typically recommend horizontal or vertical airflow fans. Watering techniques, I know out of the legacy market, hand watering, an incredible skill, and people did it with patience. But one of the challenges that we have with hand watering is there is that opportunity for splash up. And so then there's a chance that you can get things like Alternaria, Rhizoctonia to splash up onto the leaf surface, and then you've got a fungal problem. So we try to go to a bottom irrigation system, which I'll speak to a little soon. And then air purification is very critical. We're often recommending photocatalytic oxidizers that will send out charged particles of air that will attract some bacteria and fungal spores and be able to kill them. Other people are also using UV or ozone or HEPA filters within their ductwork, making sure that you are producing a good, clean air. And then, as I mentioned, nutrition is so critical. When I think of really the role of any farmer or a master grower, I really feel like their role is to optimize photosynthesis. And it's one of the most interesting things we see when we go and do some consulting is how many people are inducing so much stress on their plants because they recognize that stress is going to increase the potential for a plant to produce cannabinoids or flavonoids or terpenoids. Those secondary metabolites are a byproduct of stress, but it's so critical that we create a foundation where the plant is optimizing its photosynthesis, and we are so far beyond that, behind on that aspect. Most plants probably are working at maybe 20 to 30% of their genetic potential when it comes to being able to photosynthesize. So we're really not capturing the best of our light, temperature, CO2, and plant nutrition. So that's why I think the real role of a master grower is to make sure that we are growing the healthiest plants possible. And if we're optimizing our photosynthesis, then our plants are able to fully make complete proteins, and that's a big part of making sure you got the right form of nitrogen. When they do that, then your plants are going to have enough resources to actually store energy, and that stored energy means that when there is stress, and we will induce stress in our plants, that they have the ability, they have the resources to actually produce the flavonoid that they need, or the cannabinoid that they need, or the terpenoid that they need to address the stress. So, and this plant health pyramid, I don't know how many people are familiar with the work of John Kempf at Advancing EcoAg, but a great resource to really learn a lot more about regenerative agriculture. The other aspects I want to talk about in terms of making sure that you can have more consistent potency results is also looking at our management system. So in this soil-based system, you can see that it probably could have used a bit more refined pruning. And in a system where we're looking at probably thinking that the top 8 to 10 inches would be the ideal zone of light penetration. Anything sort of below that most likely is not going to get the same amount of light. And so a nicer prune would have created pretty much a sweet zone where you're going to have very consistent results. The interesting thing about organic, and as long as you got a well-mixed soil, things like humic acid, worm castings, they help deliver a very balanced nutrient to the plant. So even if you had perhaps a slightly larger amount of nitrogen got in somewhere, because you've got worm castings and humic acid or fulvic acid in the soils, they kind of hold on to those nutrients. When the plant needs them, then the plant will be able to take them. And so you get a more balanced uptake of nutrients in using a soil system. In a soil system, you really do want to emphasize the roots. Some chemovars will actually still, if they're making a great relationship with the plant, they still will send about 50% of their exudates into the root system, slightly more if they have an association with mycorrhizal inoculants. One of the things that we really want to emphasize when we're potting up in a living soil system is that we're looking out of the rooted clones that we're looking for. We don't really want like 6 to 8 inches of roots. We want about 2 to 3 inches of roots, so that those primary roots will then be able to develop secondary roots and then root hairs. So that's the critical part about a living soil system, is that you really want to make sure that you have roots going into as many of the pockets that are available, because that's the way they're going to get nutrients. In a hydroponic system, you're fine having a lot of great primary and secondary roots, because the nutrients are so soluble, you don't need as much surface area. And it's also important to recognize that things like biology can strongly influence root morphology. I had mentioned Bacillus Subtilis. That's another great way of having a microbe that is going to increase the mass and density of roots. I mentioned the importance of sub irrigation. So water in general is incredibly important. To have good quality water, you really want to have dissolved oxygen levels above 10 ppm. We'd like to see the temperature around 18 degrees Celsius, not cooler than 15, not warmer than 20. The reason we like sub irrigation is that we know that plants prefer to get water from below. We will either use these auto pot systems in which each plant has their own reservoir, or a capillary mat, as you see in the above picture. And one of the interesting things about using this system is, one, you're not going to get that splash up, and two, you get to create a top 2 inches of your soil that's not going to have moisture, and that'll dry out nicely. Typically, we'll get rid of any issues around fungus gnats, and the potential for other bacteria or fungi to create spores is less likely. And then, of course, I had mentioned sanitation. The importance of sanitation is critical. We do recommend shield to many of our facilities. Many of them are looking to be organic. Hydrogen peroxide is an approved substance within the Canadian organic regime. So we know that it is being used in facilities that are certified organic. And then, whether you've got a portable dram fogger or something like that, or just a backpack electrostatic sprayer, it's important to fog in and around it so that you can really get to as many surfaces as possible. But sanitation is pretty critical in trying to reduce your microbial loads. So the next point, cannabis plants grown in nutrient-dense living soils are more hardy and more robust in the presence of pests and pathogens than those plants fed with soluble nutrients. What's really, it's exciting. And sometimes you may feel like this is new to just the cannabis world, this whole idea of microbes, but this is actually relatively new to the agricultural world as well. So what we're learning here for cannabis is simultaneously being learned for horticulture and agriculture as well. So we're learning so much more about what's happening with microbes and their relationship to plants. What's interesting here is that the plant does get to dictate a lot of who hangs around the plant. So you can have, whether it's your compost that you add to your super soil or a compost tea, you could be adding thousands and thousands of species. What happens is the plant then, because of what it exudes from its roots, could be a particular amino acid, could be a terpene, could be a particular sugar, it will then be able to attract and filter out a whole bunch of other microbes. So now, instead of having thousands, maybe you've brought it down to a few hundred different species that might hang around. And then the plant will further decide what it needs when it needs it. And so then there will be certain bacteria and fungi that might be allowed to infect the plant, or that might, in fact, actually enter the plant. So the plant has a lot of control of who it wants and who it doesn't want. And so we have to learn to respect that as well. And we'll talk more about that a little later. One of the interesting things, as I was just mentioning, what we thought we knew even a decade ago, we're starting to learn a lot more. And one of the interesting things that has come up actually through my following of John Kempf, and then learning about James White's work. James White comes out of Rutgers and was talking about the rhizophagy cycle. So essentially, in this system which is evident in all plants, the root tip becomes a source where microbes are in fact engulfed by the plant. So this is almost a way similar to how ants might be farming aphids. This is a way how plants might be farming microbes. They come in through the root tip, then they are exposed to a lot of oxygen, and they essentially lose their cell membrane, release their contents. The plant gets to use their nutrients. The bacteria without its cell membrane is spit out using root hairs, and that's why with plants that are grown in a living soil, you'll see them having a lot more root hairs. These bacteria are spit out through the root hairs. Then they get all the nutrients and whatever they need to re-establish a cell membrane, and then they repeat that cycle again. So it's a way of the plant to be able to cycle a lot of nutrients, and it's a way of plants to allow for microbes to enter in, because much like our own digestive system, plants are often maintaining a lot of these microbes to perform other functions within the plant. And that's what we essentially call endophytes. So in this diagram, you can see that in the blue lettered, those are representing bacteria that are in the plant, and the red is representing fungi that are in the plant. So these aren't bacteria and fungi that are on the plant. If they're on the plant, they're called epiphytes. If they're in the plant, they're called endophytes. So these are ones that were extracted out of cannabis. You can see that there's a lot of bacteria below ground, and then there's a lot of fungi above ground. So when it comes to the addition of these microbes in a living soil system, we can go the kind of narrow down, very precise way, and that would be a very reductionist approach. So, for example, when an example would be when a species will help fix nitrogen for a plant. So we will inoculate our legumes with a particular rhizobia. Then if there's other functions, so that's nitrogen fixation, but what if we wanted something that was to protect the roots? Then we would add something that's more of an antibiotic. And so that would be what we consider stacking. And then there's the whole approach of a consortia. And the consortia idea is that we'll put out the 10,000 species or a larger, a significant amount of different species, and then allow the plant to decide what it really needs. So that would be more of an example of when you're using a compost, vermicompost, or a compost tea, and then allowing the plant to decide what it really needs. I'm just going to go over a few in particular ones that I personally think are things that we can be using in the cannabis space. One is definitely a mycorrhizal inoculant. Oftentimes people just equate mycorrhizae as an opportunity to gain phosphorus. It does do a great job of that. It's a fungi. It has the ability to solubilize phosphorus and then transport it. But it's really important to recognize that it does so much more. So mycorrhizal inoculants and associations, plants making associations with mycorrhizae, are able to transport water, are able to transport a whole bunch of other nutrients. They can access a lot of trace minerals. And what's really very interesting to watch is that once the mycorrhizae forms an association with the plant, it's now seeing itself as part of the plant. And so its goal is to make sure that the plant survives. So if there's a fusarium that is turning on the plant and is going to infect the plant, the mycorrhizal associations have the ability to attract species of bacteria that can then attack the fusarium. So it's a very interesting dynamic relationship that the plants have. In this picture here, I've highlighted a Green Star product called Synergy. The species that's used in here is called Glomus iranica. It is one that was isolated from the salt beds, I believe, of Spain. The benefit of that is that oftentimes in our living soil mixes, we can tend to run our sodium levels relatively high. We can tend to find that our EC and soluble salts is relatively high. So having a mycorrhizal inoculant that forms an association with cannabis and can tolerate salt environments is a real benefit. Some other mighty microbes, I've already mentioned Bacillus Subtilis, and there are lots of different strains of Bacillus Subtilis. Some are really good at cycling phosphorus and nitrogen, while others are good at being antagonists to pathogens, and we've used those strains as biopesticides. Trichoderma, the particular species that's in root shield, does a great job of adding root protectant. Pseudomonas species, many act as root protectants, and then some are able to solubilize iron and reduce iron to a form that the plant can use it. And lactic acid bacteria. So these are sort of facultative anaerobes. One of the byproducts of lactic acid bacteria is hexanoic acid. And hexanoic acid is a precursor to olivetolic acid, which, when it's combined with geranyl pyrophosphate, creates CBG. So you'll often see people experimenting with using lactic acid bacteria as a bit of a tea or an application, a soil drench in early flowering to try to stimulate increased cannabinoid production. So that whole concept of using teas is something that many of these super soils folks will use. Oftentimes there's enough nutrients to carry a plant into flowering, but then they want to be able to add a little bit more to give it a boost. One of the challenges that you'll find is that soluble nutrients can often create a nutrient lockout, because the plant's not used to being able to access nutrients that easily. So things like a aerobic compost tea or a fermented tea, sprouted seed teas, and even some biodynamic preparations could work well in terms of stimulating increased plant growth. There's something highlighted, don't be a moron. And I take that from Leighton Morrison. I don't know how many people are following Leighton, but a great advocate, very knowledgeable around living soils. And I think that's so true, because in our society, and definitely in the cannabis space, we often have that mentality that if something is good, then more is better. But when you're making these teas, one, I think it's really important that you do it in a very scientific way, and one that you're testing your teas, making sure that you're getting the desired species that you want, and then making sure that you understand the importance that the plant is often spending a lot of energy to address some of our management techniques. So, for example, here in this study, they were showing how adding certain exogenous applications of, in this case, hexanoic acid, can actually prime the plant and enhance the defense of the plant, right? So we're using techniques to prime the plant, almost like a vaccination. It's induced systemic resistance into the plant. What's important to recognize that if we continue to add things like a compost tea or this hexanoic acid, every time the plant is spending that energy to prime itself, you are expending energy. And so the plant is expending energy to do what it is, to react to what you've done to it. So we do need to understand how we have to time our applications, so that we're not causing a deficiency in that plant. Lastly, I just wanted to talk a little bit about how cannabis has co-evolved or evolved with microbes to provide the necessary nutrients, not only for optimal growth, but plants also recruit microbes for the production of secondary metabolites for plant production. So really important to recognize that the majority of those species kind of have a neutral relationship with the plant. So you can have thousands of species in compost, vermicompost, or in your soil, and they're really just there, part of the ecosystem, not necessarily directly or indirectly affecting the plant. But then you have some that definitely are, and what they do is that they can often induce a systemic resistance in your plant. So they trigger the plant to become primed, ready to face whatever they face. Alternatively, there's ways where you can have a pathogen either land on the leaf surface or under the roots, and it will stimulate other pathways that will kind of get the plant to produce things like cannabinoids and terpenoids. And so the two pathways that I'm most familiar with are the jasmonic acid pathway and the salicylic acid pathway, both which can be tripped and triggered by using abiotic stresses as well. Here's an example of a recent paper in which they showed how endophytes within the plant, so these are microbes within the plant that can address a pathogen that lands on it. So, for example, in this study, they were just highlighting how a plant that received aspergillus, so black mold, onto the leaf was able to use a fungal endophyte to address the pathogen that landed on the leaf. And so this is another reason why we want to have early associations with microbes to the cannabis plant, so that they can create these endophytes, or that they can bring in these endophytes, so that when you actually do get a disease, they'll have a response mechanism for that. And just to kind of wrap up, I did want to finish on a slide just to talk about the importance of looking at our organic living soils and not just assuming that those are in fact sustainable. In a regenerative agriculture process, we want to be trying to do things to mitigate climate change. And peat moss, although regulated and so-called sustainably harvested, it is still, we are harvesting it at a rate that it cannot be replaced. So we do have to start looking at substitutes for peat moss. And whether cocoa is that substitute, there's a long distance of transportation for that. Perlite is not overly sustainable in terms of the amount of heat it requires to have it created. So we have to be looking at alternatives. And I'd like to see us relying a lot more on worm castings and compost. And there's the opportunity to use composting, in-vessel composting techniques, where you can take your spent soil and within weeks be able to generate something that you could re-amend and bring back into the space. I think with that, I'm going to turn it back to Troy and hopefully have a little bit more of a discussion going.
Awesome. Thank you very much. That was extremely informative, and I hope everyone was able to take a couple of things from that. I know I was. There are some questions. I'll start off with the first one. So, do you think there is any chance of growth hormone or antibiotic residue being left over and transferred to your plants from amendments such as blood meal or manures?
That's a great question. So one of the real benefits of a soil system is that they have the ability to kind of clean up a lot of things. Now, typically, blood meal is something that is going to go through a sterilization process and break down. Sometimes people will use things like composted hen manure in there. So ideally, it should be free of things like E. coli, Salmonella, Shigella. But there could be residues of pesticides in products. There could be residues of antibiotics. A good soil is going to be able to break down a lot of that material, and that's one of the beauties of having microbes. So one of the main things that I try to recommend in all my design super soils is a fair amount of good worm castings. And those worm castings are going to start working on those materials. Either they're going to tie them in a chelated form, so they're going to hold them. They'll hold these larger macromolecules to the point where they start breaking them down, or they will actually start breaking them down. We are starting to recognize the importance of super soils in being able to, in fact, clean up a plant. So plants that may have been treated with things like myclobutanil, right? So you're using Eagle 420 or some other systemic fungicide, pesticide. One of the benefits that you can have is that plants growing in a soil over time will actually rid the plant of those those ingredients. Something that's really important, I guess right now we're starting to see a lot of interest in challenges with hop latent viroid. My thought process around a living soil system is that, and with any plant that's being overly vegetatively propagated, so we're using clones and we keep taking clones and clones, invariably we're going to get viroids and viruses in those plants. Oftentimes, what we can do best is to try to keep those asymptomatic. So as humans, we probably have viruses a lot as well, and they don't express themselves unless our immune system goes down. So one of the goals that we try to do with a healthy plant and having great photosynthesis is that we keep our immune systems in check, and then we're able to really keep that virus at bay. So the virus remains within our bodies or within the plant, but then is not expressing itself. So
it's almost like having a good gut bacteria as a human.
Yeah, great point. I think we do need to recognize that for our own our own immune system is really tied into our gut microbiome. The majority of our serotonin and dopamine is produced in the gut. We need to be feeding our gut microbiome a diverse diet, and that's what we're kind of looking for in these nutrient-dense soils. Right? It's not just NPK and a little bit of calcium, magnesium, but it's got the zinc, the cobalt, the molybdenum, the selenium, and so on.
It's very interesting. So another question, you kind of touched on this during your presentation, but I think it's a good question. So have you ever heard of any organic amendments or fertilizers being linked to failed testing in dried cannabis flowers?
Yeah, so not in terms of microbes. I've never had it tied into a microbial failing, but there is some concern around levels of arsenic and occasionally cadmium. So what's really important if you're in the regulated sector is that it's might be enough for you to just get a heavy metal analysis of a finished soil. So if you were to buy a Gaia Green product, Primal Earth, or Living Soil, make sure you ask for a heavy metal analysis of that soil. If you are designing your own soil, it's really important for you to get a heavy metal analysis of each ingredient that you want. If you're looking at your kelp meal, you want to be able to say, okay, that one does have a little bit of arsenic. Now, the arsenic in kelp, for the most part, is a organic form of arsenic. It's an arsinosugar, and is not really going to. It's not considered as toxic as an inorganic form. But most of the testing that we do in the labs here cannot differentiate between organic and inorganic arsenic, and therefore you may end up having a higher level of arsenic. Some of the rock powders may be high in cadmium and lead, so you really want to make sure that you check to see that you have relatively low rates of those products. But that's the main thing. It's really critical that you do your due diligence and you do check up on the ingredients.
For sure, and I see a lot of growers doing the same with other soils they're getting as well, or pretty much all inputs. It's a good idea to get a C of A or to test for any potential contaminants.
Right, and the same reason with things like cocoa and peat. We've often associated them as sterile media, but now we're recognizing a lot of it coming out of the work of Zamir Punja out of Simon Fraser, highlighting the fact that many of those soil media will often have fusarium and verticillium and perhaps even pythium. And so in my opinion, it's really important for us to, that's one of the reasons we want to use things like vermicompost, compost teas, is so that we can repopulate it with more beneficial microbes that can try to establish a better balance.
Yeah, it makes sense. A little bit more control. So another question, has anyone seen or looked into symbiotic relationships with different cannabis, chemovars, and specific fungi or bacteria?
You know, I think some of the work when you look at a product like Mammoth Peat, which, as far as I know, you can't access it here in Canada, but in the U.S., Mammoth Peat is a product that is I think 4 or 5 different species in it that were selected from the cannabis plant that can offer the ability to solubilize phosphorus and have some other root protectant benefits as well. So when we look at our product at Green Star, the Synergy, it is a mycorrhizal inoculant that was harvested from cannabis plants in a particular area. I know that A&L Labs is coming out with a line of microbial products. Many of those have been isolated from cannabis plants growing in particular conditions. So that's what I'm talking about is the future, I think, in terms of microbes, biostimulants, is that we're going to get to a point where we can have very designer microbes that are geared towards very specific things in the life cycle of the plant. And as we grow those plants and then recognize that perhaps in the 3rd week of flower, we recognize that the microbes around the rhizosphere and perhaps even endophytes within the plant are very different than the microbes that were around in early veg or in late flower. So we want to be able to isolate those particular microbes and be able to deliver them when the plant needs them at the rate that they want them.
Very cool. Okay, so we have about 3 more questions. I think we have enough time to get to all of them. So regarding mycorrhizae, does higher phosphorus after associating reduce efficacy, or is it just during the infection stage that low phosphorus is recommended?
Yeah, so a great point. And so the plant initially, if you have high phosphorus initially, the plant will go through that a little bit of a debate in its head whether it's going to make that association. It's important to recognize that about 40%, if you've got a great inoculation, a great infection of mycorrhizae, you may get about 40% of your exudates going into that mycorrhizae. So it's got to be a pretty important trade to be able to allow that to happen. And that's why we do recommend trying to get an early infection if you really want to have that association. Once you have an infection, the high phosphorus will only limit the spread of the mycorrhizae. The mycorrhizae may not work, that they've already got the infection into the plant. They may not go into those particular areas where you have high phosphorus levels, but they will function in different ways by transporting water, providing some root protection, accessing trace minerals and whatnot. So it's important to get that early infection, although you do see that some brands will recommend repeated application. I think the efficacy in a 8-week or 13-week plant, if you don't have early infection, you probably shouldn't be applying it later than in that early vegetative phase.
Okay, I think that should have answered that one. So another quick one. Are endophytes going to be a problem in microbial contamination analysis?
Yeah, great question. And one of the challenges that we have is our current microbial testing. It's not very species specific. It's we're not using strictly DNA-based testing, and so there are a lot of beneficials that will trigger a failed response. There is the potential of things like trichoderma, for example. Trichoderma is a root protectant. Certain species will easily be translocated that could potentially create a failed microbial. I'm not aware of it, of course, because we don't really break it down to a species level, so we really don't know what's causing it. I think for the most part, I mean, that's probably why certain biopesticides have not been permitted, because if you use those biopesticides, you'd invariably get a failed microbial test. Yeah, so I definitively, I don't think I can say that the endophytes would be the cause. I think the numbers that you would get would be far lower than what you'd have if you had an exogenous microbial population.
For sure. Okay, so can you speak on the importance of sulfur on plant health and potential terpene/cannabinoid production, also gypsum as a source for said sulfur?
Great. Yeah. I mean, to me, sulfur is pretty much synonymous with anything around aromatics. So if you're looking for terpene production, you definitely want to have good sulfur. Sulfur remains pretty critical when it comes to the synthesis for a lot of proteins as well. So, along with a lot of those trace minerals, having sulfur as a bit of a catalyst to get some protein synthesis happening is integral. So having adequate amount of sulfur is great. Then we definitely look at different sources of sulfur. So gypsum is a great source of sulfur. Epsom salts, magnesium sulfate, another good source, and then sulfomag, K-mag is also a real good source. The benefit of using something like a gypsum is the fact that it doesn't alter your pH, and so you get some calcium and you get the sulfur. The main thing you want to be able to do is, with a lot of these rock powders, is make sure you've got a fine, not a pelletized form. You want it in a fine flour because you want to increase that surface area. Typically, any of these rock powders, they give off more as they get weathered away. Having a good fungal component within your super soil is definitely going to help break down rock powders quicker and releasing those beneficial nutrients.
Question from me: would using warm water for that help?
Not really. It's so if you had a pelleted form of something, then warm water might help help it dissolve better. Mm-hmm. But these powders actually need to be kind of broken down through acids that are secreted by fungi. We use a weathering. That's why we would typically recommend lining in the fall, because the winter will work away at the limestone and make it more available. So another approach is making sure that you've got active microbial life that will break apart and start working on these nutrients. Perfect.
So this is the last question we have. If anyone else has any other questions that you didn't get a chance to write down, Av's information is there. So while using LABs in early flower, would this be used as a soil drench?
Yeah. So often sources, if you're making your own labs, typically I kind of, because we're working in almost two different ecosystems, we've got a aerobic compost tea compost system with that's kind of got a whole slew of its own microbial population. And then when we talk about labs, we're starting to talk about facultative anaerobes. And so I try to keep them slightly separate, unless you're going in a strict, sort of natural farming mentality, in which you are going to use a lot of more fermented fermentation type teas and fermentation systems, like effective microorganisms or indigenous microorganisms. So I try to separate the two. And so if I choose to go into using labs, I'm going to do that in the flowering because I know the benefits of making sure that I have hexanoic acid. And so I would use it at that time and maybe use either compost teas and aerobic tea thing early in the plant's life, and then go towards it perhaps late in the plant's life. So in the middle part, perhaps we can do a little testing, and I mean, a lot of this is still in that research phase in which you want to be able to test and see what is actually giving you the result that you desire.
And that is actually all the time we have for today. So thank you so much to you, Av, for such an amazing presentation. And thank you to everybody for joining us today. It's always great to see such a lively chat. If you have any further questions, please visit shield.ca. And you will also receive a follow-up email with a link to the recording of today's webinar. So keep an eye on your inboxes. Thank you again for joining us, and we hope to see you on our next webinar.
Thank you.
Thank you all. Thank you.